enhance the transcription of target genes. They are vital for the regulation of gene expression, acting as switches that determine whether a gene should be turned on or off, depending on cellular signals, environmental stimuli, or developmental stage. These proteins do not directly bind RNA polymerase but act through protein-protein interactions and chromatin modifications to facilitate the assembly and function of the transcriptional machinery, so recruiting the transcriptional machinery to the correct. They are typically modular in nature, with distinct domains responsible for DNA binding and activation. This lecture covers the following key areas. Structure of transcription activators. DNA binding domains and activation domains. Mechanism of action of activators. Role in chromatin remodeling. Interaction with coactivators and mediator. Enhancers and activator binding sites. Examples of transcription activators. Previous year questions with answers. 1. Structure of transcription activators. Transcription activators generally consist of two functional domains. DNA binding domain (DBD). This domain recognizes and binds to specific DNA sequences. Enhancers or upstream regulatory elements. Activation domain (AD). This domain interacts with coactivators, chromatin remodeling complexes, or general transcription factors to enhance transcription. Some activators may have additional domains for signal sensing, e.g., ligand binding domain and nuclear receptors dimerization, or regulatory control. 2. DNA binding domains and activation domains. Common DNA binding domains include helix turn helix HTH found in homeodomain proteins. Zinc finger motifs seen in steroid hormone receptors. Leucine zipper BZIP found in proteins like CREB. Helix loop helix VHLH found in transcription factors such as MYD. Activation domains. Acidic activation domains found in yeast activators like GAL4 and BP16. Glutamine-rich domains found in SP1. Proline-rich domains found in CTF-NF1. These domains do not directly contact the basal transcription machinery but work through protein-protein interactions. 3. Mechanism of action of activators. Transcription activators increase gene expression by several mechanisms. Recruitment of coactivators. They bind coactivators like CBP P300 which possess histone acetyltransferase activity, loosening chromatin. Interaction with the mediator complex. Activators help recruit the mediator, which bridges activators and RNA polymerase II. Stabilization of the pre-initiation complex picked by recruiting GTFs and RNA pol II to the promoter. Chromatin modification. Indirectly promote transcription by altering histone structure through recruitment of remodeling complexes. Activators may work synergistically, where two or more activators together lead to much higher transcription than each alone. 4. Role in chromatin remodeling. DNA is packed into chromatin, which can repress transcription. Activators recruit chromatin remodeling complexes like SWI-SNF, which use ATP to reposition nucleosomes, making DNA accessible. They also recruit histone acetyltransferases (HATs) to acetylate histones and reduce their affinity for DNA, making the promoter region more open. 5. Interaction with coactivators and mediator. Coactivators do not bind DNA directly but are recruited by activators. Some key coactivators include CBP P300, histone acetyltransferases that modify chromatin to an open state. Mediator complex, a multi-subunit complex that physically links activators with RNA polymerase II and GTFs. Mediator acts as an integrator of signals and assists in the formation and stabilization of the transcription initiation complex. 6. Enhancers and activator binding sites. Activators bind to enhancers, which may be located upstream, downstream, or even within the genes they regulate. These enhancers work irrespective of orientation or distance due to DNA looping, allowing activators to interact with the promoter-bound transcription machinery. Enhancer-promoter communication is facilitated by the looping of DNA, bringing distant regulatory elements in close proximity with the core transcription machinery. 7. Examples of transcription activators. GAL4 East recognizes UASG sequences, activates transcription via acidic activation domain. NF-kappa B activates immune and inflammatory genes upon stimulation. CREB, activated by phosphorylation via CAMP signaling, binds to CAMP response elements. P53, a tumor suppressor that activates genes involved in DNA repair, apoptosis, and cell cycle arrest. Estrogen receptor ER, activated by hormone binding, binds to estrogen response elements ARES. These transcription activators often respond to external signals and integrate intracellular pathways to fine-tune gene expression. 8. Previous year questions with answers. Question: CSIRNET 2022. Which of the following is true regarding transcriptional activators in eukaryotes? A. They bind RNA polymerase II directly at the core promoter. B. They bind to top box in the promoter region. C. They bind to enhancer regions and facilitate transcription via coactivators. D. They unwind DNA at the transcription start site. Answer: C. They bind to enhancer regions and facilitate transcription via coactivators. Question: CSIRNET 2020. Which of the following is a function of the acidic activation domain of a transcriptional activator like GAL4? A. DNA methylation. B. Direct RNA synthesis. C. Histone acetylation via recruitment of coactivators. D. DNA replication. Answer: C. Histone acetylation via recruitment of coactivators. Question: CSIRNET 2019. Which of the following statements about transcription activators is incorrect? A. They bind to specific DNA sequences. B. They interact with the basal transcription machinery. C. They have histone methyltransferase activity. D. They often possess acidic or proline-rich activation domains. Answer: C. They have histone methyltransferase activity. Correct explanation: transcription activators themselves do not have histone methyltransferase activity. They may recruit such enzymes indirectly. Conclusion: transcription activators are key regulators of gene expression in eukaryotic cells. They perform their function by binding specific regulatory sequences, recruiting coactivators and chromatin remodeling complexes, and facilitating the assembly and activation of the transcription machinery. Their modular architecture allows flexibility and adaptability in gene regulation. Understanding their mechanisms, domains, and interactions is essential for cracking CSIRNET JRF life sciences, as this area is frequently tested and holds significant conceptual weight. You said. Transcription repressor. ChatGPT said. Transcription repressors. CSIRNET JRF life sciences. Detailed lecture. Maximum detail. Note table. PYQs included. Transcription repressors are regulatory proteins that inhibit gene expression by interfering with the process of transcription. While transcription activators promote transcription by enhancing the recruitment and function of RNA polymerase and general transcription factors, repressors prevent transcription through multiple molecular strategies. These mechanisms are crucial for maintaining cell type specificity, temporal gene expression, developmental control, and response to environmental signals. Understanding transcription repressors is essential for the CSIRNET JRF life sciences exam, especially given that questions often test knowledge of gene regulatory mechanisms, protein DNA interactions, chromatin structure modulation, and the function of specific repressor pathways. 1. Definition and importance. A transcription repressor is a DNA binding protein that negatively regulates gene expression by blocking the transcription of target genes. It binds to specific DNA sequences, often overlapping or adjacent to the promoter or enhancer regions, thereby preventing the access or function of the transcriptional machinery. This ensures that genes are expressed only when required, contributing to energy conservation and regulatory precision. 2. Types of transcription repressors. Repressors can be classified based on their mode of action. A. Competitive repressors. They bind to DNA sequences that overlap with or are adjacent to activator binding sites, thereby competing with activators for binding. This direct competition prevents the recruitment of coactivators or general transcription factors. B. Masking repressors. Quenching. They do not block DNA binding of activators but bind directly to the activation domains of activators, thereby preventing them from interacting with the transcriptional machinery or coactivators. C. Direct repression through core repressor recruitment. They recruit core repressor complexes such as histone deacetylases, HDACs, histone methyltransferases, or other chromatin modifying enzymes, which compact chromatin and make it inaccessible for transcription initiation. D. Repressor mediated chromatin condensation. They may actively recruit chromatin remodeling complexes that compact nucleosomes and silence gene expression, such as polycomb group proteins (PCG). 3. Mechanisms of action: blocking RNA polymerase recruitment. Repressors can bind to operator or silencer regions near the promoter to prevent the assembly of the pre-initiation complex (PIC) or block the recruitment of RNA polymerase II. Preventing enhancer promoter communication. By binding to insulator elements or silencing elements, repressors can prevent looping of DNA, thereby disrupting enhancer promoter communication required for transcription activation. Modulating chromatin state. Repressors recruit chromatin modifying enzymes. Histone deacetylases (HDACs) remove acetyl groups from histones, causing tighter DNA histone interaction and transcriptional repression. Histone methyltransferases (HMTs) methylate histones to generate repressive marks like H3K9Me3 or H3K273, which are associated with heterochromatin. Histone methyltransferases (HMTs) methylate histones to generate repressive marks like H3K9Me3 or H3K273, which are associated with heterochromatin. Nucleosome remodeling complexes. Compact chromatin or reposition nucleosomes to cover the transcription start site. 4. Silencer elements. Silencers are specific DNA sequences to which repressors bind. These elements can act at a distance and in an orientation-independent manner, much like enhancers. When bound by repressors, they inhibit transcription by preventing chromatin opening, PIC assembly, or enhancer function. 5. Examples of transcription repressors: REST and RSF, Re1 silencing transcription factor, represses neuronal genes in non-neuronal tissues by recruiting chorus and HDACs. MAD-MAX complex. Opposes MYC-MAX activation by recruiting SYN3 core repressor and HDACs. Groucho TLE family, in Drosophila invertebrates, recruited by repressor proteins to mediate gene silencing during development. Snail and slug, zinc finger repressors. Suppress E-cadherin expression during epithelial to mesenchymal transition, EMT, essential in development and cancer metastasis. Polycomb repressive complexes, PRC1 and PRC2, epigenetically silence genes through H3K27 methylation and chromatin compaction. 6. Transcriptional core repressors. Core repressors are non-DNA binding proteins that are recruited by repressors to execute repression. Examples include NCOR and SMRT, core repressors of nuclear hormone receptors that recruit HDACs. SYN3A, recruited by many repressors including MAD, REST, and MECP2. CTBP, C-terminal binding protein, interacts with many repressor proteins and brings HDACs and other chromatin modifiers. These multiprotein complexes often form large repressive assemblies on chromatin to silence transcription stably inheritably. 7. Biological significance. Developmental regulation ensures genes are turned off in inappropriate tissues, e.g., REST represses neuronal genes in non-neuronal tissues. Cell cycle control. Repressors like RB protein block expression of S-phase genes. X chromosome inactivation, mediated by long non-coding RNAs and chromatin-associated repressor complexes. Cancer biology. Mutations in repressors like P53 or deregulation of repressive chromatin marks are linked to tumorigenesis. 8. Previous year questions with answers 1. CSIR Net Life Sciences 2021. Which of the following is a true mechanism by which a transcriptional repressor can inhibit gene expression? A direct methylation of the promoter DNA B activation of histone acetyltransferase activity C binding to enhancer elements to facilitate activator function D recruitment of histone deacetylases to the promoter region. Answer. D recruitment of histone deacetylases to the promoter region. Explanation. This is a classical mechanism of repression wherein HDACs remove acetyl groups, leading to chromatin condensation and transcriptional repression. 2. CSIR Net Life Sciences 2019. REST Re1 silencing transcription factor represses neuronal gene expression in non-neuronal cells by activating transcription factor CREBB recruiting coactivators to histone acetylate nucleosome C binding to promoter and recruiting histone deacetylases D promoting enhancer promoter communication. Answer. C binding to promoter and recruiting histone deacetylases. Explanation. REST binds to Re1 sites and recruits chorus and HDACs to silence neuronal genes outside the nervous system. 3. CSIR Net Life Sciences 2020. Which of the following protein complexes is associated with transcriptional repression? A SWI-SNFB mediator CCBP P300. D polycomb repressive complex. Answer. D polycomb repressive complex. Explanation. PRC1 and PRC2 are known to repress gene expression by methylating H3K27 and compacting chromatin. Conclusion. Transcription repressors are indispensable to the precise control of gene expression, acting through DNA binding competition, masking activators, recruiting core repressors, and remodeling chromatin to a repressive state. Their roles span development, differentiation, metabolism, and disease, making them high-priority targets in transcriptional regulation questions for CSIR Net JRF Life Sciences. A strong grasp of their mechanisms, examples, and differences from activators is crucial for cracking conceptual and applied questions in the exam. You said, RNA polymerases. ChatGPT said, RNA polymerases. CSIR Net JRF Life Sciences. Detailed theory lecture. Maximum detail based on PYQ areas. No table includes PYQs with year and answers. RNA polymerases are essential enzymes responsible for the synthesis of RNA from a DNA template in a process known as transcription. These enzymes read the DNA template strand in the 3' to 5' direction and synthesize a complementary RNA strand in the 5' to 3' direction. In both prokaryotes and eukaryotes, RNA polymerases are central to gene expression, but their structure, number, and functions differ significantly between the two domains. Understanding RNA polymerases is crucial for the CSIR Net JRF Life Sciences exam because transcription is a fundamental process in molecular biology, and questions frequently test candidates on types of polymerases, their roles, inhibitors, subunit composition, promoter recognition, and associated factors. 1. RNA polymerase in prokaryotes. Prokaryotic organisms, such as Escherichia coli, have a single type of RNA polymerase responsible for transcribing all classes of RNA, mRNA, tRNA, and rRNA. Structure. Prokaryotic RNA polymerase is a multi-subunit enzyme. The core enzyme has 5 subunits. Alpha 2 copies involved in enzyme assembly and interaction with regulatory proteins. Beta, catalytic subunit responsible for RNA synthesis. Beta binds to the DNA template. Omega assists in enzyme assembly and stability. To initiate transcription, the sigma factor joins the core enzyme to form the holoenzyme, which can recognize and bind specific promoter sequences. Function of sigma factor. It directs the holoenzyme to the promoter by recognizing conserved sequences like the -10 prid box and -3 5 regions. After initiation, the sigma factor dissociates, and the core enzyme proceeds with elongation. Different sigma factors recognize different classes of genes under various conditions, providing regulatory specificity. 2. RNA polymerases in eukaryotes. Eukaryotic cells contain 3 primary nuclear RNA polymerases, each with distinct roles. A RNA polymerase I. coli, located in the nucleolus, transcribes a single gene type, the large rRNA precursor 45S, which is processed into 18S, 5.8S, and 28S rRNAs. B. RNA polymerase II, Pol2, located in the nucleoplasm, transcribes protein coding genes mRNA and also synthesizes snRNAs and some RNAs. Requires several general transcription factors GTFs for initiation, e.g., TFIID, TFIIH. Associated with CTD, C-terminal domain of the largest subunit RPB1, which undergoes phosphorylation to regulate initiation and elongation. C. RNA polymerase III, Pol3, located in the nucleoplasm, transcribes tRNAs, 5S rRNA, and other small RNAs. Each of these polymerases recognizes different promoter elements and utilizes distinct transcription factors to initiate transcription. 3. Mitochondrial and chloroplast RNA polymerases. These organelles possess bacteria-like single subunit RNA polymerases that are similar to T7 bacteriophage polymerase. They transcribe mitochondrial or chloroplast genes and are nuclear encoded but function within the organelle. 4. RNA polymerase structure and catalytic mechanism. RNA polymerases share a conserved catalytic core characterized by a crab claw-like shape. The enzyme forms a transcription bubble, approximately 17 base pairs, by unwinding DNA. Nucleoside triphosphates (NTPs) are added one by one to the 3' of the grown RNA chain, and pyrophosphate is released with each addition. The enzyme progresses with high processivity and fidelity, although proofreading is limited compared to DNA polymerases. The Mg² ion is essential for catalysis, coordinating the phosphate groups of NTPs. 5. Inhibitors of RNA polymerases. Actinomycin D intercalates DNA and blocks elongation by all polymerases. Rifampicin specifically inhibits bacterial RNA polymerase by binding the beta subunit and preventing transcription initiation. Used against Mycobacterium tuberculosis. Alpha-amanitin, a toxin from Amanita phalloides mushroom. Inhibits eukaryotic RNA polymerase II at low concentrations and RNA polymerase III at higher doses. Pola is resistant. These inhibitors are often exam favorites in CSIR-NET due to their specificity and practical applications in research and medicine. 6. Regulation of polymerase activity. RNA polymerase activity is modulated by transcription factors, chromatin structure, post-translational modifications, especially CTD phosphorylation in POL2, and interactions with enhancers silencers. The transition from initiation to elongation in POL2 is a highly regulated step involving kinases like TFIIH, which phosphorylates CTD, promoting promoter clearance. 7. Differences between RNA polymerase and DNA polymerase. RNA polymerase does not require a primer. It can initiate synthesis de novo. It lacks exonuclease proofreading activity, making RNA more error-prone. DNA polymerases require a template and primer, and they have higher fidelity with proofreading. Previous year questions with answers 1. CSIR-NET Life Sciences 2018. Which one of the following toxins specifically inhibits eukaryotic RNA polymerase II? A rifampicin B actinomycin D C alpha-amanitin D cyclohexamide. Answer: C alpha-amanitin. Explanation: Alpha-amanitin inhibits RNA polymerase II at nanomolar concentrations, making it a potent tool in transcription studies and a common exam question. 2. CSIR-NET Life Sciences 2020. In eukaryotic cells, which RNA polymerase is responsible for the transcription of tRNA genes? A RNA polymerase IB, RNA polymerase II, circa RNA polymerase III, D mitochondrial RNA polymerase. Answer: cRNA polymerase III. Explanation: RNA pol 3 transcribes tRNAs, 5S rRNA, and small nuclear RNAs. 3. CSIR-NET Life Sciences 2021. Which subunit of bacterial RNA polymerase recognizes the promoter region of DNA? A beta B beta, C alpha D sigma. Answer: D sigma. Explanation: the sigma factor is essential for promoter recognition and binding in prokaryotes. It is released after initiation. Conclusion: RNA polymerases are central to the gene expression machinery in all living organisms. Prokaryotes utilize a single enzyme with sigma factors for specificity, whereas eukaryotes possess 3 distinct polymerases, poli, 2, 3 each dedicated to transcribing specific types of genes. Understanding their structure, function, specificity, and inhibitors is critical for the CSIR-NET JRF Life Sciences exam. Mastery of this topic ensures clarity in transcriptional regulation and builds a solid foundation for related topics like transcription factors, initiation complex formation, and RNA processing. You said, capping, ChatGPT said, capping. CSIR-NET JRF Life Sciences, complete theory lecture, maximum detail based on previous year questions, no table includes PYQs with year and answers. Capping is a vital co-transcriptional modification that occurs at the 5' end of nascent pre-mRNA transcripts in eukaryotic cells. This modification results in the formation of a unique structure known as the 5' cap, which is essential for mRNA stability, nuclear export, efficient translation, and protection from exonucleases. The process of capping occurs almost immediately after the RNA polymerase II transcribes around 20 to 30 nucleotides of the mRNA. It is an indispensable quality control and regulatory step in eukaryotic gene expression and is not seen in prokaryotes. 1. Definition and structure of the 5' cap. The 5' cap is a modified guanine nucleotide attached to the first nucleotide of the RNA transcript via a 5' to 5' triphosphate linkage, which is chemically unusual and resistant to nucleases. This cap is further methylated at the N7 position of the guanine base, resulting in 7-methylguanosine M to the power of 7G. In some cases, the ribose sugars of the first and second nucleotides are also methylated at the 2' position, forming cap 1 and cap 2 structures respectively. 2. Steps in the capping process. Capping involves 3 major enzymatic steps that occur co-transcriptionally. RNA triphosphatase activity. The enzyme removes the gamma phosphate from the 5' triphosphate end of the nascent RNA, leaving a diphosphate end. Ganylyltransferase activity. This enzyme adds GMP, guanosine monophosphate, from GTP to the diphosphate end of the RNA through a 5' to 5' triphosphate linkage, creating the basic cap structure. Methyltransferase activity. The enzyme methylates the guanine at the N7 position, converting the cap to 7-methylguanosine M to the power of 7G, which is the hallmark of a functional 5' cap. Additional methylations may occur on the ribose sugars of the first and second nucleotides. 3. Enzyme complex and CTD involvement. The enzymes involved in capping are recruited by the phosphorylated C-terminal domain (CTD) of RNA polymerase II. Specifically, the SR5 phosphorylated CTD serves as a platform for assembling and positioning the capping machinery. This coordination ensures that capping is tightly coupled with transcription. 4. Biological functions of the 5' cap of protection from degradation. The 5' cap protects pre-mRNA from degradation by 5' exonucleases, increasing transcript stability. B. Facilitates nuclear export. Capping is required for the binding of cap binding complex (CBC), which mediates nuclear export of mRNA through the nuclear pore complex. C. Promotes translation initiation. In the cytoplasm, the cap is recognized by A4E, a component of the eukaryotic initiation factor complex, which helps recruit the ribosome to the mRNA for cap-dependent translation initiation. D. Splicing efficiency. The presence of a cap enhances the efficiency of splicing of the first intron, indicating a role in coordinating multiple RNA processing steps. E. Quality control. Uncapped mRNAs are rapidly degraded, ensuring only properly processed mRNAs are translated. 5. Cap binding proteins. In the nucleus, the cap binding complex (CBC) binds the 5' cap and interacts with splicing and export machinery. In the cytoplasm, the 5' cap is recognized by A4E, which is essential for ribosome recruitment and translation initiation. The decapping enzyme complex DCP1 DCP2 removes the cap during regulated mRNA decay pathways like nonsense-mediated decay (NMD) and general mRNA turnover. 6. Cap-independent translation. Some viral RNAs and certain eukaryotic mRNAs initiate translation without a 5' cap, using internal ribosome entry sites (IRIS). These are exceptions to the standard cap-dependent initiation. 7. Differences between capping in eukaryotes and prokaryotes. Capping is exclusive to eukaryotes. Prokaryotic mRNAs do not undergo capping. Instead, they possess a triphosphate at the 5' end and are rapidly translated, often co-transcriptionally, without the need for cap structures. Previous year questions with answers 1. CSIR-NET Life Sciences 2017. Which of the following modifications occurs at the 5' end of a eukaryotic mRNA? Addition of poly A tail B removal of intron C addition of 7-methylguanosine demethylation of 3' end. Answer: C addition of 7-methylguanosine. Explanation: the hallmark of 5' capping is the addition of a 7-methylguanosine residue in a 5' to 5' linkage to protect and regulate the mRNA. 2. CSIR-NET Life Sciences 2020. Which of the following statements about the 5' cap structure in eukaryotic mRNA is correct? A. It is added after transcription is complete. B. It promotes mRNA degradation. C. It is recognized by A4E during translation initiation. D. It is found in prokaryotic mRNAs. Answer. C. It is recognized by E4E during translation initiation. Explanation. The 5 feed cap is critical for translation initiation in eukaryotes, where A4E binds the cap and recruits the translation machinery. 3. CSIR Net Life Sciences 2019. The enzyme that adds a guanine residue to the 5 feed end of eukaryotic mRNA during capping is a poly A polymerase B ganylyltransferase cRNA helicase D spliceosome. Answer. B ganylyltransferase. Explanation. This enzyme catalyzes the transfer of GMP to the 5 feed end of the RNA forming the cap structure. Conclusion. Capping is an essential and unique feature of eukaryotic mRNA processing that plays a central role in the regulation of gene expression at multiple levels: transcription, splicing, export, translation, and stability. It represents one of the earliest quality control steps for RNA transcripts. Mastery of this topic is important for cracking CSIR Net Life Sciences, as questions frequently test both mechanistic understanding and functional consequences of RNA capping. Be sure to correlate this topic with RNA polymerase II, CTD phosphorylation, and translation initiation factors for an integrated understanding. You said, elongation, ChatGPT said, elongation of transcription. CSIR Net JRF Life Sciences, complete theory lecture, maximum detail, previous year questions included, no tables, paragraph format. Transcription elongation is a central phase of gene transcription during which RNA polymerase travels along the DNA template, synthesizing a complementary RNA strand by adding ribonucleotides in a 5' to 3' direction. Following initiation and promoter clearance, RNA polymerase transitions into a process of elongation complex. This phase is highly regulated and plays a critical role in ensuring transcriptional fidelity, efficiency, and responsiveness to various cellular signals. 1. Transition from initiation to elongation. After the formation of the transcription initiation complex and synthesis of the first few nucleotides, typically around 10, RNA polymerase undergoes a conformational change, losing most general transcription factors in eukaryotes or sigma factor in prokaryotes. This allows it to escape the promoter and enter a stable elongation phase. In eukaryotes, this transition is highly dependent on phosphorylation of the RNA polymerase II C-terminal domain (CTD), particularly at SR2 residues, which allows recruitment of elongation factors in RNA processing machinery. 2. Mechanism of elongation. During elongation, RNA polymerase unwinds the DNA helix ahead of it to expose the template strand and rewinds it behind, forming a structure called the transcription bubble, typically 14 to 17 base pairs long. Within this bubble, about 8 to 9 nucleotides of the newly formed RNA remain base paired to the DNA template. The enzyme catalyzes the formation of phosphodiester bonds between the 3' group of the grown RNA chain and the incoming ribonucleoside triphosphates, RNTPs, which are selected based on Watson-Crick base pairing rules with the DNA template. 3. Elongation factors in eukaryotes. In eukaryotic cells, several elongation factors associate with RNA polymerase II to regulate elongation rate, pausing, and RNA processing. Key among them are TFIIS, resolves transcriptional pausing and assists in RNA cleavage to allow backtracked RNA polymerase to resume. DSIF-DRB, sensitivity inducing factor, and NELF, negative elongation factor, induce early elongation pausing near promoters. This is released by the action of PTAP, positive transcription elongation factor B, which phosphorylates RNA pol 2 CTD, SR2, DSIF, and NELF. SPT5, SPT6, promote transcriptional elongation and chromatin reorganization. PTAP CDK9 plus cyclin T, a major elongation activator, phosphorylates negative factors to allow elongation to proceed. 4. Pausing and proofreading. Transcription elongation is not always continuous. RNA polymerase can pause due to DNA sequences, chromatin structures, or incorporation errors. Pausing allows for co-transcriptional RNA processing like capping, and serves as a regulatory checkpoint. Paused polymerase may either resume elongation or undergo backtracking, in which case the 3' end of the RNA is displaced from the active site. This requires transcript cleavage factors like TFIIS to realign the active site and allow elongation to resume. 5. Supercoiling and topoisomerases. As RNA polymerase progresses, it introduces positive supercoiling ahead and negative supercoiling behind the transcription bubble. This torsional stress must be resolved by topoisomerases, topo I and 2, which relieve supercoiling and facilitate smooth progression of elongation. 6. Chromatin and nucleosome dynamics. In eukaryotes, RNA polymerase must transcribe through nucleosomal DNA. This is achieved with the help of chromatin remodelers and elongation factors like FACT, facilitates chromatin transcription, which disassemble and reassemble nucleosomes. Histone modifications, such as acetylation, also make chromatin more accessible to polymerase during elongation. 7. Co-transcriptional RNA processing. As elongation proceeds, capping, splicing, and eventually polyadenylation signals are added co-transcriptionally. The CTD of RNA polymerase II acts as a platform to recruit and coordinate these RNA processing machineries, making transcription elongation a hub of gene expression regulation. 8. Elongation in prokaryotes. In bacteria, the sigma factor is released after initiation, and core RNA polymerase continues elongation. Elongation factors like NUSA and GRIA assist in pausing and proofreading. Bacterial RNA polymerase also faces supercoiling challenges and pauses due to terminator sequences or regulatory proteins. 9. Transcription elongation and gene regulation. Recent studies show that elongation is a major regulatory point in gene expression. In metazoans, many genes are transcribed but held in a paused state until specific signals, e.g., stress or developmental cues, release RNA polymerase to complete transcription. This phenomenon is particularly common in developmentally regulated and inducible genes. Previous year questions with answers 1. CSIR Net Life Sciences 2016. What is the role of PTAP in transcription elongation? A inhibits elongation by binding RNA pol 2. B enhances elongation by phosphorylating CTD and DSIF-C cleaves backtracked RNA. D acetylates histones ahead of RNA pol 2. Answer. B enhances elongation by phosphorylating CTD and DSIF. Explanation. PTAP phosphorylates SR2 of the CTD, as well as DSIF and NELF, converting paused RNA polymerase into a productive elongation complex. 2. CSIR Net Life Sciences 2018. Which of the following statements about eukaryotic transcription elongation is false? A TFIIS stimulates cleavage of nascent RNA to correct misincorporation. The RNA polymerase II CTD is phosphorylated during elongation. C. Elongation is assisted by FACT complex for chromatin traversal. D. Elongation occurs after translation in eukaryotes. Answer. D. Elongation occurs after translation in eukaryotes. Explanation. In eukaryotes, transcription and translation are spatially and temporally separated, and elongation occurs before translation in the nucleus, whereas translation occurs in the cytoplasm. 3. CSIR Net Life Sciences 2021. Which factor facilitates transcription elongation through nucleosomes? A Grub B FACT circuit TFIIDDU1SNRNP. Answer. B FACT. Explanation. FACT facilitates chromatin transcription by temporarily displacing histone H2A, H2B dimers and reassembling nucleosomes after the passage of RNA polymerase II. Conclusion. Transcription elongation is not merely a passive extension of initiation, it is a highly dynamic, regulated, and coordinated phase of gene expression. It involves a complex interplay of elongation factors, chromatin remodelers, topoisomerases, and RNA processing proteins, all of which ensure accuracy, responsiveness, and proper maturation of transcripts. For CSIR Net JRF Life Sciences, understanding the molecular players and events in elongation is vital, especially in integrating it with gene regulation and co-transcriptional events like capping and splicing. You said, termination, ChatGPT said. Termination of transcription. CSIR Net JRF Life Sciences, complete theory lecture, maximum detail based on previous year questions, no tables, paragraph format. Termination of transcription is the final phase in the transcription process during which RNA polymerase ceases RNA synthesis, releases a newly synthesized RNA molecule, and dissociates from the DNA template. This step is crucial for defining the 3' end of the transcript and for recycling the polymerase for subsequent rounds of transcription. Termination mechanisms vary between prokaryotes and eukaryotes, and understanding these distinct mechanisms is fundamental to gene regulation. 1. Transcription termination in prokaryotes. In bacteria, there are two main mechanisms of transcription termination. A row-independent intrinsic termination. Rho-independent termination is mediated purely by specific sequences within the DNA. The transcribed RNA contains a GC-rich palindromic sequence followed by a stretch of uracils. The GC-rich region forms a stable hairpin structure, stem loop, in the RNA, which causes RNA polymerase to pause. The U-rich sequence that follows forms a weak RNA-DNA hybrid due to AU base pairs, leading to destabilization of the transcription complex and release of the RNA transcript. B. Rho-dependent termination. This mechanism requires a Rho factor, a hexameric ATP-dependent RNA helicase. Rho binds to a Rho utilization site on the nascent RNA, which is AC-rich, G4, an unstructured region. It translocates along the RNA in a 5' to 3' direction using ATP hydrolysis. When RNA polymerase pauses at a termination site, Rho catches up and unwinds the RNA-DNA hybrid, causing the release of RNA and dissociation of the complex. 2. Transcription termination in eukaryotes. Eukaryotic transcription termination is more complex and polymerase-specific. RNA polymerase I termination. Termination of RNA poli transcription involves a termination factor called TTFI, transcription termination factor for poli, which binds to specific terminator sequences downstream of rRNA genes and induces pausing of the polymerase. Additional factors such as PTRF, polymerase I, and transcript release factor help dissociate the transcription machinery from the DNA. RNA polymerase III termination. RNA pol III terminates transcription at a short stretch of thymine residues, T tracks, in the DNA template. Unlike pol I and pol II, pol III does not require additional termination factors, and the RNA transcript is released immediately after the polymerase encounters the poly T sequence. RNA polymerase II termination. Pol II termination is the most elaborate and involves coupling with pre-mRNA 3' end processing. There are two main models for pol II termination. Torpedo model. After transcription of the polyadenylation signal AAUAA, the pre-mRNA is cleaved by cleavage and polyadenylation factors such as CPSF and CSTF. The upstream RNA is polyadenylated and processed, while the downstream RNA, still attached to pol II, is rapidly degraded by 5' to 3' exonuclease called XRN2 in humans. XRN2 catches up with pol II and induces its release from DNA. Allosteric conformational change model suggests that transcription of the polyadenylation signal causes a structural change in the RNA polymerase or the transcription complex, reducing its processivity and leading to dissociation. Most likely, both models act in combination, where a conformational change allows XRN2 to terminate the polymerase effectively. 3. Role of polyadenylation signals in termination. In pol II transcription, polyadenylation signals e.g., AAUAA and downstream GU-rich sequences are critical for both 3' end processing and transcription termination. The process involves recruitment of cleavage factors, CPSF, CSTF, poly A polymerase, and other proteins. The coupling of cleavage and termination ensures that only full-length, properly processed transcripts are released. 4. Transcription termination and gene regulation. Termination is a regulatory checkpoint ensuring RNA molecules are full-length and ready for further processing. Defects in termination can lead to read-through transcription, transcriptional interference with downstream genes, and genomic instability. Moreover, alternative polyadenylation APA at different cleavage sites can result in mRNAs with varying 3' UTRs, affecting stability, localization, and translational efficiency, thus influencing gene expression regulation in development, differentiation, and disease. 5. Transcription termination in mitochondria. Mitochondrial transcription termination also involves unique factors like mTOR, mitochondrial transcription termination factor. These proteins bind to specific DNA sequences downstream of transcription units to pause or terminate transcription in a strand-specific manner. 6. Termination and recycling of RNA polymerase. After termination, RNA polymerase is recycled for another round of transcription. In bacteria, core RNA polymerase rebinds to sigma factor to initiate new transcription. In eukaryotes, dephosphorylation of the pol II CTD is necessary before it can be reused, which is carried out by CTD phosphatases like FCP1. Previous year questions with answers 1. CSIR Net Life Sciences 2015. Which of the following is required for Rho-dependent termination in prokaryotes? AAGC-rich hairpin in RNA-BA poly T sequence in DNA C-Rho protein and red site depolyadenylation signal. Answer: C-Rho protein and red site. Explanation: Rho-dependent termination requires the Rho helicase to bind the red site on RNA and disrupt the RNA-DNA hybrid using ATP hydrolysis. 2. CSIR Net Life Sciences 2017. Which of the following best describes the torpedo model of transcription termination in eukaryotes? A cleavage of RNA by RNase HB binding a Rho factor to DNA. C 5' to 3' exonuclease mediated degradation of RNA leading to polymerase release of RNA at poly T tract. Answer: C 5' to 3' exonuclease mediated degradation of RNA leading to polymerase release. Explanation: In the torpedo model, XRN2 degrades the RNA downstream of the cleavage site and catches up to pol II, triggering termination. 3. CSIR Net Life Sciences 2020. What is the role of the polyadenylation signal in RNA pol II termination? A pausing of ribosomes during translation B facilitates 5' capping C recruitment of termination and cleavage factors D enhances mRNA nuclear export. Answer: C recruitment of termination and cleavage factors. Explanation: The polyadenylation signal is recognized by CPSF and CSTF, which mediate RNA cleavage and recruit polyadenylation machinery, also contributing to transcription termination. Conclusion: Termination of transcription is not merely the end of RNA synthesis but an integral part of gene regulation and transcript maturation. Its diversity among different RNA polymerases and domains of life reflects its evolutionary importance. Understanding the molecular mechanisms behind both prokaryotic and eukaryotic termination, as well as their regulatory implications, is essential for mastering gene expression at the level required to excel in CSIR Net JRF Life Sciences. This knowledge also connects deeply with mRNA processing, stability, and the overall control of gene output in response to cellular demands. You said, RNA processing. ChatGPT said, RNA processing. CSIR Net JRF Life Sciences. Full-length theory lecture paragraph format, no tables, maximum detail based on previous year questions. RNA processing is a crucial set of post-transcriptional modifications that precursor RNAs, pre-RNAs undergo to become functionally mature RNA molecules in both prokaryotic and eukaryotic cells, though it is more elaborate and regulated in eukaryotes. This includes capping, splicing, editing, polyadenylation, trimming, and chemical modifications, which collectively determine the stability, localization, and translatability of RNA transcripts. While prokaryotic mRNAs are usually translated directly after transcription with minimal processing, eukaryotic primary transcripts, especially pre-mRNAs, require extensive modifications before they can be transported to the cytoplasm and translated. 1. mRNA processing in eukaryotes. A 5' capping. The addition of a 7-methylguanosine cap at the 5' end of nascent pre-mRNA is the first modification. The capping process involves the removal of the terminal phosphate by an RNA triphosphatase, addition of GMP by dinylyl transferase, and methylation of the guanine at the end 7 position by methyltransferase. This cap protects mRNA from exonucleases, facilitates nuclear export, and is recognized by cap-binding proteins involved in splicing and translation initiation. B. Splicing. Pre-mRNA splicing is the removal of non-coding introns and joining of exons by the spliceosome, a large ribonucleoprotein complex consisting of small nuclear RNAs, SNR NOS U1, U2, U4, U5, U6, and associated proteins. The spliceosome assembles a conserved intronic sites, 5' splice site, branch point A, polypyrimidine tract, and 3' splice site, and catalyzes 2 transesterification reactions. Alternative splicing generates multiple mRNA isoforms from a single gene, increasing proteomic diversity. Splicing errors can lead to exon skipping or intron retention, often associated with diseases. C 3' polyadenylation. After the cleavage of the pre-mRNA at a specific site downstream of the polyadenylation signal AAUAA, a poly A tail of approximately 200 adenines is added by poly A polymerase. This process is mediated by a complex of cleavage and polyadenylation factors including CPSF, CSTF, CFIM, CFIIM, and PAP. The poly A tail enhances mRNA stability, nuclear export, and translation efficiency by interacting with poly A binding proteins PABPs. 2. RNA editing. RNA editing is a post-transcriptional process that alters the nucleotide sequence of RNA, thus changing the coding potential of the transcript. It includes substitution editing, such as adenosine to inosine (ADAR) editing by ADAR enzymes, and cytidine to uridine (CDU) editing by APOBEC enzymes. Editing can alter codons, splice sites, or RNA secondary structure, thereby influencing gene expression and protein function. A classical example is the editing of apolipoprotein B, a POB mRNA, where a CDU change introduces a premature stop codon, generating two isoforms, APOB100 and APOB48. 3. RNA splicing variants and regulation. Alternative splicing is a highly regulated process influenced by cis-acting elements, exonic, intronic splicing enhancers or silencers, and trans-acting factors SR proteins and HNRNPs. These interactions determine which exons are retained in the final mRNA. Regulation of splicing is crucial during development, cell differentiation, and stress responses. Misregulation is linked to diseases such as spinal muscular atrophy and cancer. 4. rRNA and tRNA processing. Pre-rRNA transcripts 45S in humans undergo cleavage, base modification, methylation, and pseudoregulation, and assembly with ribosomal proteins to form mature 18S, 5.8S, and 28S rRNAs. These modifications are guided by small nucleolar RNAs, SNRNAs. tRNA processing includes cleavage by RNase P, 5' end, and RNase C, 3' end addition of CCA sequence at the 3' terminus, and extensive base modifications e.g., methylation, thiolation, inosine formation critical for accurate decoding during translation. 5. mRNA surveillance and quality control. Eukaryotic cells possess RNA quality control mechanisms such as nonsense-mediated mRNA decay, NMD, non-stop decay, NSD, and no-go decay, NGD, to eliminate defective transcripts. NMD targets mRNAs with premature stop codons. NSD degrades mRNAs lacking stop codons. NGD clears transcripts with stalled ribosomes. These pathways ensure only functional RNAs are translated. 6. RNA transport and localization. Processed RNAs are selectively exported from the nucleus through the nuclear pore complex. mRNA export requires cap binding complex, CBC, X junction complex, EJC, and TAP NXF1 transport receptors. Specific RNAs are also localized within the cytoplasm for spatial regulation of translation, a process particularly important in neurons and embryonic development. 7. Small RNA processing, mRNAs and siRNAs. MicroRNAs, mRNAs, and small interfering RNAs, siRNAs, are processed from precursor transcripts by Drosha and Dicer, and then incorporated into RNA-induced silencing complex RISC. These small RNAs guide RISC to target complementary mRNAs to repress translation or induce degradation, playing crucial roles in post-transcriptional gene silencing and defense against viruses. 8. RNA processing in prokaryotes. Although less extensive, prokaryotes also process rRNA and tRNA. A polycistronic precursor transcript is cleaved by RNases such as RNase 3 and RNase P to generate individual rRNAs and tRNAs. tRNAs undergo further trimming and base modifications, though mRNA processing is minimal due to the absence of introns and nucleus. 9. Coupling of transcription and processing. In eukaryotes, RNA processing is tightly coupled to transcription by RNA polymerase II. The C-terminal domain (CTD) of RNA pol II acts as a scaffold to recruit processing factors during transcription. Phosphorylation of CTD regulates the sequential recruitment of capping enzymes, splicing factors, and polyadenylation complexes. This ensures precise and efficient RNA maturation. 10. Biological significance and disease association. Defective RNA processing is linked to several genetic diseases. For example, mutations in spliceosomal components can lead to retinitis pigmentosa, and errors in polyadenylation have been associated with beta thalassemia. Aberrant editing and splicing patterns also contribute to cancer progression. Therefore, understanding RNA processing is vital not only for basic molecular biology but also for disease diagnostics and therapeutics. Previous year questions with answers 1. CSIR Net Life Sciences 2016. Which of the following statements is true regarding RNA splicing? A. It only occurs in prokaryotes. B. It removes exons from the transcript. C. It is mediated by the spliceosome in eukaryotes. D. It adds a poly A tail to mRNA. Answer. C. It is mediated by the spliceosome in eukaryotes. Explanation. The spliceosome removes introns and joins exons to form mature mRNA in eukaryotes. 2. CSIR Net Life Sciences 2019. A mutation that alters the AAUAA sequence in eukaryotic pre-mRNA would most likely affect a transcription elongation B splicing. C. Capping D polyadenylation. Answer. D polyadenylation. Explanation. The AAUAA sequence is the canonical signal for 3' end cleavage and polyadenylation in eukaryotic mRNAs. 3. CSIR Net Life Sciences 2017. Which of the following RNAs is processed by both RNase P and tRNA nucleotide transferase? A. mRNA B tRNA C rRNA D mRNA. Answer. B tRNA. Explanation. RNase P processes the 5' end of tRNA, while tRNA nucleotide transferase adds the 3' CCA tail necessary for amino acid attachment. Conclusion. RNA processing is an indispensable aspect of gene expression regulation in eukaryotic cells. It ensures that RNA molecules are correctly modified, matured, and sorted for their respective cellular roles. Mastering the mechanisms and regulation of capping, splicing, editing, polyadenylation, and surveillance is essential for success in CSIR Net JRF Life Sciences. This knowledge connects core molecular biology to developmental biology, disease mechanisms, and biotechnology. You said, RNA editing. ChatGPT said, RNA editing. CSIR Net JRF Life Sciences. Comprehensive lecture, maximum detail, paragraph format, no tables, includes PYQs with answers in year. RNA editing is a post-transcriptional modification process by which nucleotide sequences of an RNA molecule are altered after transcription, leading to a final RNA sequence that differs from the genomic DNA template. It plays a vital role in expanding transcriptomic and proteomic diversity and fine-tuning gene expression in various organisms. Unlike RNA splicing, which removes introns, RNA editing changes specific nucleotides within the RNA molecule, thereby modifying its coding potential, stability, localization, or secondary structure. Types of RNA editing. The two major types of RNA editing in eukaryotes are substitution editing and insertion deletion editing. 1. Substitution editing. This is the most common form in higher eukaryotes, involving the chemical conversion of one base to another. Adenosine to inosine (ADI) editing. This type is catalyzed by a class of enzymes called ADARs, adenosine deaminases acting on RNA. Inosine is interpreted as guanosine during translation and by the splicing machinery. This form of editing primarily occurs in the coding regions and untranslated regions (UTRs) of mRNA. ADI editing can create new splice sites, alter codons e.g., converting glutamine codon to arginine, and modify microRNA binding sites. Cytidine to uridine (CDU) editing. This occurs through APOBEC, apolipoprotein B mRNA editing catalytic polypeptide-like enzymes. A well-known example is the editing of apolipoprotein B mRNA, where a CAA codon is converted to UAA, a stop codon producing a truncated protein APOB48 in the intestine compared to the full length APOB100 in the liver. 2. Insertion deletion editing. This is primarily observed in the mitochondria of kinetoplastic protozoans like trypanosoma. It involves the insertion or deletion of uridine U residues into the mRNA and is guided by guide RNase (GRNAs). These GRNAs base pair with free mRNA and direct the editing machinery to add or delete uridines at specific sites, thus generating a functional open reading frame. This type of editing can be extremely extensive and is essential for the expression of mitochondrial genes. Mechanism of RNA editing. ADI editing mechanism. The editing site is often within a double-stranded RNA structure formed by the exon or intronic complementary sequences. ADAR enzymes bind to these cs-RNA regions and catalyze the hydrolytic deamination of adenosine to inosine. Since inosine pairs like guanosine, this leads to codon changes and affects protein structure and function. ADAR1 and ADAR2 are the two main isoforms involved in mammals. Editing specificity is determined by RNA secondary structure and sequence context. CDU editing mechanism. APOBEC1 binds to a Morin sequence near the editing site and deaminates cytidine to uridine. Accessory proteins like ACF, APOBEC1 complementation factor, stabilize this complex and determine site specificity. Uridine insertion deletion mechanism in trypanosomes. The GRNA binds to the pre-mRNA at the editing site and forms a mismatch. The mismatch guides the endonuclease to cleave the mRNA, followed by uridine insertion by terminal ribotransferase 2-tase or deletion by exonuclease. Finally, RNA ligase rejoins the edited RNA fragments. Multiple rounds of editing are often required for full transcript maturation. Biological functions and importance. RNA editing significantly increases transcriptome plasticity without altering the DNA sequence. In the nervous system, ADI editing affects ion channels, neurotransmitter receptors e.g., AMPA and serotonin receptors, and synaptic plasticity. Editing of GLUR mRNA, for instance, changes a glutamine to an arginine, altering calcium permeability of AMPA receptors and protecting neurons from excitotoxicity. RNA editing also influences innate immunity, apoptosis, development, and circadian rhythms. Dysregulation of editing is linked to diseases like epilepsy, depression, glioblastoma, amyotrophic lateral sclerosis, ALS, and hepatocellular carcinoma. In plants, RNA editing is common in mitochondrial and chloroplast transcripts, mostly C to U changes. These edits restore codons for conserved amino acids and functional proteins essential for respiration and photosynthesis. Regulation of RNA editing. RNA editing is tightly regulated in a cell type specific, developmental stage specific, and stimulus responsive manner. Factors influencing editing include the availability and localization of editing enzymes, expression of GRNAS in kinetoplastids, RNA secondary structure, and interaction with RNA binding proteins. For instance, a DAR1 is induced by interferons and plays a role in modulating immune responses. Deficiency of a DAR1 results in unedited dsRNAs triggering MDA5 mediated innate immune activation. RNA editing versus RNA splicing. While both processes alter the mRNA post-transcriptionally, RNA splicing removes introns and joins exons to generate mature mRNA, whereas RNA editing changes the nucleotide content without cutting out RNA segments. Importantly, RNA editing can even alter splice sites and thereby indirectly influence splicing patterns. RNA editing in human health and disease. Alterations in RNA editing patterns are increasingly recognized as biomarkers and therapeutic targets. Reduced editing of GLUR mRNA is associated with epilepsy and gliomas. Hyperediting can lead to overproduction of immunostimulatory dsRNA, promoting autoimmune diseases like Aicardi-Goutieres syndrome. Some cancers exhibit editing-dependent activation or silencing of tumor suppressor or oncogenic transcripts. Thus, the RNA editing landscape serves as a critical layer of gene regulation, with potential for precision medicine. Previous year questions PYQs 1. CSIRNet Life Sciences 2015. In the intestinal cells, apolipoprotein B mRNA is edited to produce a shorter version of the protein. This is achieved by AATI editing by DAR BC to U editing by APOBDC alternative splicing detranslational pausing. Answer: BC to U editing by APOBDC. Explanation: the C to U RNA editing by APOBDC1 converts CAA glutamine to UAA stop codon truncating the protein into a POB48. 2. CSIRNet Life Sciences 2018. Which of the following is true about ADAR enzymes? A. They convert cytosine to uracil. B. They recognize single-stranded RNA. C. They act only on ternus. D. They convert adenosine to inosine. Answer: D. They convert adenosine to inosine. Explanation: ADARs are RNA-specific adenosine deaminases that act on double-stranded RNA regions and deaminate adenosine to inosine. 3. CSIRNet Life Sciences 2021. Insertion deletion type RNA editing is primarily seen in a human nuclear mRNA B's mitochondrial RNA C trichanosome mitochondrial RNA D bacterial mRNA. Answer: C trichanosome mitochondrial RNA. Explanation: in trichanosoma, RNA editing involves guide RNAs that direct uridine insertion or deletion in mitochondrial transcripts. Conclusion: RNA editing is a dynamic, sequence-specific, and enzymatically driven alteration of RNA nucleotides that broadens genetic information without modifying the genome. It represents a sophisticated post-transcriptional regulatory layer that is critical for normal cellular function and adaptive responses. Mastering the mechanisms, enzymes involved, and biological implications of RNA editing is essential to cracking the CSIRNet JRF Life Sciences exam, as it is a frequently tested and conceptually rich topic that integrates molecular biology, developmental biology, and disease biology. You said, splicing, ChatGPT said, RNA splicing. CSIRNet JRF Life Sciences, high yield, maximum detail lecture, paragraph format, note tables, PYQs with year and answer. RNA splicing is a post-transcriptional process in eukaryotes wherein non-coding introns are precisely removed from the pre-mRNA, primary transcript, and protein-coding exons are ligated to form a continuous mature mRNA. This process is a critical aspect of gene expression regulation and is essential for the generation of functional transcripts that can be translated into proteins. It occurs in the nucleus and is orchestrated by a large ribonucleoprotein complex known as the spliceosome. Constitutive and alternative splicing. There are two primary forms of RNA splicing. Constitutive splicing involves the removal of all introns and joining of exons in the order they appear. Alternative splicing allows for different combinations of exons to be joined together, producing multiple protein isoforms from a single gene. This dramatically increases the proteomic complexity without requiring additional genes and is particularly prevalent in higher eukaryotes, especially in neural and immune tissues. Spliceosome and its components. The spliceosome is a dynamic complex composed of 5 small nuclear ribonucleoproteins, SNRNPs, U1, U2, U4, U5, and U6, and numerous associated proteins. Each SNRNP contains a small nuclear RNA, SNRNA, that guides the splicing reaction through base pairing with specific sequences on the pre-mRNA. The splicing reaction is directed by consensus sequences found at the junctions of introns and exons. 5' splice site, donor site typically begins with GU. 3' splice site, acceptor site typically ends with AG. Branch point site, usually an adenine A, located upstream of the 3' splice site within a conserved sequence. Polypyrimidine tract, rich in C and U nucleotides and found near the branch point. Mechanism of splicing: two-step transesterification reaction. Step 1: the 2' of the branch point attacks the phosphate at the 5' splice site, forming a lariat structure with a 2' minus 5' phosphodiester bond and releasing the 5' exon. Step 2: the free 3' of the 5' exon then attacks the 3' splice site phosphate, ligating the 2 exons together and releasing the intron in its lariat form. This reaction is catalyzed by the rearrangement and interactions of SNRNPs. U1 SNRNP recognizes the 5' splice site, U2 binds the branch point, and U4 U6 U5 tri-SNRNP is recruited to form the active spliceosome. After catalysis, the spliceosome disassembles and the SNRNPs are recycled. Alternative splicing patterns. Alternative splicing enables a single gene to produce multiple transcripts, and its types include exon skipping, mutually exclusive exons, alternative 5' or 3' splice sites, intron retention. This process is regulated by cis-acting elements, exonic, intronic splicing enhancers or silencers, and trans-acting factors like SR proteins, splicing enhancers, and HNRNPs, splicing repressors. Misregulation of these elements can cause diseases like cancer, spinal muscular atrophy, and myotonic dystrophy. Splicing and mRNA surveillance. The exon junction complex (EJC) is deposited near exon boundaries post-splicing. This complex plays roles in mRNA export, nonsense-mediated decay (NMD), and translation regulation. mRNAs with premature stop codons upstream of an EJC are targeted for degradation by NMD, ensuring faulty transcripts do not produce harmful truncated proteins. Self-splicing introns. Apart from spliceosomal splicing, self-splicing introns group 1 and group 2 exist primarily in lower eukaryotes, mitochondria, and chloroplasts. Group 1 introns utilize a free guanosine cofactor to initiate the splicing reaction. Group 2 introns follow a mechanism similar to spliceosomal splicing and are believed to be evolutionary precursors of the spliceosome. Clinical significance. Defects in splicing contribute to genetic disorders and oncogenesis. For example, mutations at splice sites can lead to exon skipping or intron retention. Mutations in SMN1 gene splicing lead to spinal muscular atrophy. Many cancers show altered splicing of tumor suppressors and oncogenes, e.g., BCLX long versus short isoforms. Antisense oligonucleotide therapy, such as Spinraza for spinal muscular atrophy, works by modifying splicing patterns to restore functional protein production. Previous year questions PYQs 1. CSIRNet Life Sciences 2016. The lariat structure formed during splicing contains a unique phosphodiester bond. It is A 2 feet minus 5 feet between branch point A and G at 5 feet splice site B 3 feet minus 5 feet between branch point A and G C 2 feet minus 5 feet between branch point G and at 3 feet and D 3 feet minus 5 feet between branch point A and U. Answer. A 2 feet minus 5 feet between branch point A and G at 5 feet splice site. Explanation. The 2 feet O of the branch point attacks the 5 feet splice site G, forming a 2 feet minus 5 feet phosphodiester bond. 2. CSIR Net Life Sciences 2018. The recognition of the 5 feet splice site in pre-mRNA is primarily done by AU2SNRNP BU4SNRNP CU1SNRNP DU5SNRNP. Answer. CU1SNRNP. Explanation. U1SNRNP base pairs with the 5 feet splice site of the intron during the early stages of spliceosome assembly. 3. CSIR Net Life Sciences 2022. Alternative splicing increases. A. The number of genes in the genome. B. The number of ternus. C. The diversity of proteins without increasing gene number. D. The replication rate of DNA. Answer. C. The diversity of proteins without increasing gene number. Explanation. Alternative splicing allows one gene to encode multiple protein isoforms, enhancing proteomic diversity. Conclusion. RNA splicing is a vital mechanism in eukaryotic gene expression that ensures the accurate glycosylation begins with the recognition of a highly conserved hexanucleotide sequence motif, AAUAA, located about 10 to 30 nucleotides upstream of the cleavage site in the 3' untranslated region, 3' UTR of the mRNA. A GU-rich or U-rich downstream sequence element, DSE, is often found 20 to 40 nucleotides downstream of the cleavage site and is essential for the efficiency of cleavage and polyadenylation. These sequence motifs are recognized by a set of protein complexes including CPSF cleavage and polyadenylation specificity factor binds the AAUAA signal. CSTF cleavage stimulation factor binds the GU-rich DSE. CFI and CFII cleavage factors I and 2 aid in the cleavage. Polyadenylation. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. Polyadenylation is a post-transcriptional RNA processing event in eukaryotes wherein a stretch of adenine nucleotides, called the poly A tail, is added to the 3' end of the nascent pre-mRNA. This event is critical for the stability, nuclear export, and translation of mRNA. The poly A tail also plays essential roles in mRNA surveillance and regulation of gene expression. It occurs co-transcriptionally, usually after the transcription of the gene is complete, and is tightly regulated by multiple protein complexes. Signal for polyadenylation. Polyadenylation begins with the recognition of a highly conserved hexanucleotide sequence motif, AAUAA, located about 10 to 30 nucleotides upstream of the cleavage site in the 3' untranslated region, 3' UTR of the mRNA. A GU-rich or U-rich downstream sequence element, DSE, is often found 20 to 40 nucleotides downstream of the cleavage site and is essential for the efficiency of cleavage and polyadenylation. These sequence motifs are recognized by a set of protein complexes including CPSF cleavage and polyadenylation specificity factor binds the AAUAA signal. CSTF cleavage stimulation factor binds the GU-rich DSE. CFI and CFII cleavage factors I and 2 aid in the cleavage reaction. PAP is the addition of adenine residues. PABP poly A binding protein binds to the poly A tail and regulates its length. Mechanism of polyadenylation. The process proceeds in 2 steps. Cleavage. The pre-mRNA is cleaved at a site 10 to 30 nucleotides downstream of the AAUAA sequence by the assembled protein complex. This cleavage site often occurs after a CA dinucleotide. Poly A tail addition. After cleavage, poly A polymerase PAP adds approximately 200 to 250 adenosine residues in mammals to the 3' end without a template. This reaction is ATP dependent. As the tail is synthesized, poly A binding protein PABP binds the grown tail and modulates the process. Functional roles of poly A tail. The poly A tail plays crucial roles in the life cycle of mRNA. mRNA stability. The tail protects mRNA from 3' right pointing arrow 5' exonucleolytic degradation. Shortening of the tail leads to mRNA decay. Translation. PABP interacts with A4G, part of the translation initiation complex, to circularize the mRNA, promoting ribosome recruitment and efficient translation. Nuclear export. Proper polyadenylation is required for the mRNA to be recognized by the nuclear export machinery and transported to the cytoplasm. mRNA quality control. Improperly processed or mutated mRNAs lacking poly A tails are retained in the nucleus or degraded. Regulation of polyadenylation. Polyadenylation is regulated developmentally and tissue specifically, especially in processes like oocyte maturation, early embryogenesis, and neuronal function. Many genes have multiple polyadenylation sites. Alternative polyadenylation or APA allowing different 3' UTR lengths, which influences mRNA targeting, translation efficiency, mRNA stability. APA contributes significantly to the post-transcriptional regulation of gene expression and proteomic diversity. For instance, cancer cells often show shifts toward proximal polyadenylation sites, leading to shorter 3' UTRs and escape from mRNA-mediated repression. Polyadenylation in histone mRNAs and prokaryotes. Unlike most eukaryotic mRNAs, replication-dependent histone mRNAs lack poly A tails. Instead, they terminate in a conserved stem loop structure, which is bound by the stem loop binding protein SLBP. In prokaryotes, polyadenylation exists but serves a different function. It targets mRNA for degradation rather than stabilizing it. Clinical relevance in poly A tail mutations. Mutations in polyadenylation signals or processing factors can result in various genetic disorders and cancers. For example, beta thalassemia can arise from a mutation in the AAUAA polyadenylation signal of the beta globin gene, resulting in aberrant or inefficient 3' end processing. Polyadenylation is also manipulated therapeutically using antisense oligonucleotides to alter poly A site selection or processing efficiency. Previous year questions. PYQs 1. CSIR Net Life Sciences 2015. Which of the following sequences is most commonly found upstream of the polyadenylation site in eukaryotic pre-mRNA? AUGUGGBAUAA circa AUUUGCDUUUAG. Answer. BAAUAA. Explanation. The AAUAA hexamer is the canonical polyadenylation signal recognized by CPSF during 3' end processing. 2. CSIR Net Life Sciences 2021. Poly A polymerase adds a poly A tail. A using a DNA template. B using an RNA template. C in a sequence-specific, template-independent manner. D is a part of ribosomal subunit assembly. Answer. C in a sequence-specific, template-independent manner. Explanation. PAP adds adenosine residues to the 3' end of mRNA without using a template. 3. CSIR Net Life Sciences 2017. What is the role of poly A binding protein PABP? A catalyzes cleavage of the pre-mRNA. B synthesizes the poly A tail. C enhances stability and translation of mRNA. D cleaves the lariat structure during splicing. Answer. C enhances stability and translation of mRNA. Explanation. PABP binds to the poly A tail, stabilizing mRNA and promoting efficient translation initiation. Conclusion. Polyadenylation is an indispensable step in eukaryotic mRNA maturation that ensures the survival, export, and translation of transcripts. The addition of a poly A tail, guided by specific sequence motifs and regulated by precise enzymatic machinery, is critical for post-transcriptional gene regulation. This process also integrates with alternative polyadenylation mechanisms to influence mRNA fate and proteome diversity. Given its central importance in mRNA metabolism and frequent testing in CSIRNet life sciences, especially in conceptual and application level questions, mastering this topic is essential for cracking the exam confidently and completely. You said. Structure and function of different types of RNA. ChatGPT said. Structure and function of different types of RNA. CSIRNet JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. RNA ribonucleic acid plays a central role in gene expression, protein synthesis, and regulation of cellular processes. Unlike DNA, RNA is single-stranded and contains the sugar ribose instead of deoxyribose. The four nucleotides in RNA are adenine A, uracil U, cytosine C, and guanine G with uracil replacing thymine. There are various types of RNA, each with distinct structures and functions essential for cellular processes. 1. Messenger RNA mRNA structure. mRNA is a linear molecule that is transcribed from DNA and carries genetic information from the nucleus to the cytoplasm for protein synthesis. It has a 5' cap added during transcription that helps protect the mRNA and assists in ribosome binding. A poly A tail at the 3' end which enhances stability, facilitates nuclear export, and aids translation. Coding regions exons that encode proteins and non-coding regions untranslated regions, UTRs, at both ends. Function. mRNA serves as a template for protein synthesis during translation. It is transcribed from genes and contains the codons that determine the sequence of amino acids in the resulting protein. 2. Transfer RNA tRNA structure. tRNA is a small, cloverleaf shaped molecule, typically consisting of 70 to 90 nucleotides. It has an anticodon loop that recognizes and binds to a complementary codon on the mRNA during translation. A 3' group that binds to an amino acid, which is specific to the tRNA type. DR and TR regions that are involved in tRNA recognition and binding to ribosomal sites. Function. tRNA acts as the adapter molecule in protein synthesis. It decodes the mRNA codons and delivers the appropriate amino acids to the ribosome, where they are incorporated into the growing polypeptide chain. Each tRNA is specific to one amino acid. 3. Ribosomal RNA rRNA structure. rRNA molecules form the structural and catalytic components of the ribosome. There are several types of rRNA in both prokaryotes and eukaryotes, with the most common being 16 srRNA in prokaryotes, small ribosomal subunit. 18 srRNA in eukaryotes, small ribosomal subunit. 23 srRNA in prokaryotes and 28 srRNA in eukaryotes, large ribosomal subunit. These rRNAs fold into complex three-dimensional structures and are stabilized by interactions with proteins. Function. rRNA serves both structural and catalytic roles in protein synthesis. It forms the scaffold for ribosomal protein assembly. Catalyzes the peptide bond formation during translation, ribozymal function. Helps align mRNA and tRNA for proper codon-anticodon matching during translation. 4. Small nuclear RNA snRNA structure. snRNA molecules are typically 100 to 300 nucleotides long and are primarily found in the nucleus. They are components of the spliceosome, a complex that facilitates RNA splicing. The major snRNAs include U1, U2, U4, U5, U6 involved in the splicing process. Function. snRNAs are essential for pre-mRNA splicing. They form complexes with proteins to create snRNPs, small nuclear ribonucleoproteins, which recognize splice sites, catalyze intern removal, and ligate exons together in the mature mRNA. 5. Small nucleolar RNA SNRNA structure. SNRNAs are small RNA molecules, typically 60 to 300 nucleotides in length, located in the nucleus. They are involved in the modification of rRNA molecules, guiding specific chemical modifications like methylation and pseudoregulation. Function. SNRNAs are responsible for the modification of rRNAs, which is essential for the proper functioning of the ribosome. They guide the enzymatic modification of rRNA nucleotides, contributing to the stability and functionality of the ribosome. 6. MicroRNA structure. mRNAs are small, approximately 20 to 22 nucleotides long and are transcribed from non-coding genes. They form a hairpin-like secondary structure before being processed by Drosha and Dicer into their mature form. Function. mRNAs regulate gene expression by binding to the 3' UTR of mRNA targets, usually resulting in mRNA degradation or translation inhibition. They play a role in the regulation of various biological processes, such as development, differentiation, apoptosis, and stress response. 7. Long non-coding RNA LNC-RNA structure. LNCRNAs are long RNA molecules greater than 200 nucleotides in length, and they do not encode proteins. They have various secondary structures and can form complex tertiary structures. Some LNCRNAs are polyadenylated, while others are not. Function. LNCRNAs are involved in regulating gene expression at multiple levels, including transcriptional regulation. Some LNCRNAs interact with chromatin-modifying complexes to influence gene expression. Post-transcriptional regulation. They can influence mRNA stability, splicing, and translation. Chromatin organization. Certain LNCRNAs are involved in the formation of nuclear domains, regulating chromatin structure and accessibility. 8. Pee-wee interacting RNA pRNA structure. pRNAs are small RNA molecules, approximately 24 to 31 nucleotides long, that interact with pee-wee proteins. They are primarily expressed in the germline and are derived from long single-stranded precursors. Function. pRNAs play a crucial role in silencing transposable elements in germ cells, thereby protecting the genome from transposon-induced damage. They also contribute to the regulation of gene expression in the germline and are involved in epigenetic regulation and the maintenance of genome stability. 9. Circular RNA SRSNA structure. SRSNAs are a special class of non-coding RNA that form a closed-loop structure, meaning they lack a 5' or 3' end. They are generated by back-splicing events, where the 3' end of an exon is joined to the 5' end of the same or a different exon. Function. While the functions of SRSNAs are still being explored, they are believed to act as mRNA sponges, regulating mRNA availability. Participate in protein scaffolding. Influence transcriptional regulation and RNA splicing. Previous year questions PYQs 1. CSIRNet Life Sciences 2016. Which of the following RNAs is directly involved in the splicing of pre-mRNA? rRNA B mRNA C tRNA dsNRNA. Answer. dsNRNA. Explanation. snRNAs are integral components of the spliceosome, which catalyzes the splicing of pre-mRNA. 2. CSIRNet Life Sciences 2019. Which RNA molecule is associated with chromatin modification and gene silencing in the germline? mRNA B LNC RNA C pRNA D SRSNA. Answer. C pRNA. Explanation. pRNAs are involved in transposon silencing and epigenetic regulation in the germline, protecting the genome from transposon activity. 3. CSIRNet Life Sciences 2017. What is the main function of the 5' cap added to eukaryotic mRNA? A protection from exonucleases. B. Initiation of translation. C. Polyadenylation. D. Splicing of introns. Answer. B. Initiation of translation. Explanation. The 5' cap is essential for ribosome binding during translation initiation, protecting the mRNA from degradation. Conclusion. RNA molecules are central to various biological processes, from protein synthesis to gene regulation. Each type of RNA has a specific structure. Each type of RNA has a specific structure and function that is crucial for cellular activities. Understanding the roles of different RNAs is fundamental to grasping how genetic information is expressed and regulated, making this topic essential for success in the CSIRNet JRF Life Sciences exam. The high relevance of this topic in both conceptual and application-based questions ensures it will be a major focus in your preparation. You said. RNA transport. ChatGPT said. RNA transport. CSIR Net JRF Life Sciences, maximum detail, paragraph format, no tables, PYQs included with year and answers. RNA transport refers to the process by which RNA molecules are moved from one compartment of the cell to another, particularly from the nucleus to the cytoplasm, where protein synthesis occurs. This is a crucial process in eukaryotic cells, as most RNA transcription takes place in the nucleus, while translation protein synthesis occurs in the cytoplasm. The efficient and regulated transport of RNA is vital for maintaining cellular functions, ensuring proper gene expression, and regulating processes like cell differentiation, response to stress, and growth. 1. RNA transport in eukaryotes. In eukaryotic cells, the nucleus and cytoplasm are separated by the nuclear envelope, and RNA must pass through the nuclear pore complexes (NPCs) to reach the cytoplasm. This process is highly regulated and involves several steps. RNA processing and export from the nucleus. Before RNA is exported from the nucleus, it undergoes several processing steps. Capping of the 5' end. Splicing to remove introns from free mRNA. Polyadenylation of the 3' end of mRNA. These modifications are crucial for the stability and transport of the RNA. Only fully processed, mature RNA molecules are exported from the nucleus. For example, mRNA is processed with a 5' cap, poly A tail, and removal of introns. Nuclear export signals NES and NLS. Proteins involved in RNA export interact with nuclear export signals NES and nuclear localization signals NLS on RNA binding proteins. For example, exportins are responsible for recognizing the NES on RNA protein complexes and guiding the complex to the nuclear pore. The NLS is recognized by importance for proteins that enter the nucleus. Transport machinery. Once the RNA is processed and bound by the appropriate proteins, it forms a ribonucleoprotein complex RNP. This complex is recognized by the nuclear pore complex NPC, a highly selective gateway. The NPC is composed of nucleophorins that form a channel for RNA and ribonucleoprotein complexes to travel through. The transport process requires energy in the form of GTP and involves several transport factors, such as CRM1, a key protein for exporting RNA and proteins. TAP-P15, a complex involved in the export of mRNA. This export process is highly regulated, and only mature, processed RNAs are allowed to pass through the NPC. Defects in RNA export can lead to various diseases, including cancer and neurodegenerative disorders. 2. mRNA export from the nucleus to the cytoplasm. Once the mature mRNA is properly processed, it is exported from the nucleus to the cytoplasm. This export is mediated by mRNA export receptors and involves the following key steps. The mRNA is bound by RNA binding proteins, including TAP and NXF1, which are essential for binding the mRNA to the nuclear pore complex. The mRNA protein complex is transported through the NPC, with GTP hydrolysis providing the necessary energy. The 5' cap structure of mRNA and poly A tail are recognized by the export machinery, ensuring the mRNA is properly transported. After mRNA reaches the cytoplasm, it undergoes further processing, such as the removal of any remaining export factors. The mRNA is then available for translation by ribosomes. 3. Turn and rRNA transport. Unlike mRNA, turn and rRNA do not undergo the same type of processing in the cytoplasm. These RNAs are synthesized in the nucleus, where they undergo processing, e.g., methylation, pseudouracylation for rRNA, and are then exported to the cytoplasm for their roles in translation. Turna, transported from the nucleus to the cytoplasm after processing, where it is loaded with amino acids and participates in protein synthesis. rRNA, synthesized and processed in the nucleus and then exported to the cytoplasm, where it forms the structural and catalytic components of the ribosome. Both turn and rRNA rely on similar export receptors as mRNA but are not necessarily subject to the same stringent regulation regarding export signals. 4. Regulation of RNA export. The export of RNA is tightly regulated at multiple levels. For mRNA, this regulation is especially important for ensuring that only properly processed and mature mRNAs are exported. Several proteins and complexes are involved in this regulation. Go complex, involved in mRNA export by interacting with the RNA and facilitating its passage through the NPC. TREX complex, associated with the Go complex and assists in mRNA export by interacting with both the RNA and the nuclear pore complex. ALI-REF complex, recognizes the mRNA cap and is essential for the export of mRNA. Additionally, the RNA itself can have regulatory sequences, such as RNA export elements, RES, and mRNA transport signals, which facilitate their recognition and export. RNA binding proteins play a crucial role in this regulation, ensuring that only fully processed, mature RNA is exported to the cytoplasm. 5. RNA reimport into the nucleus. While most RNA is transported from the nucleus to the cytoplasm, there is also evidence that RNA molecules can be reimported into the nucleus, particularly in cases of RNA surveillance and quality control. For example, mRNA decay. mRNAs that are not properly translated or have errors may be sent back to the nucleus for degradation. RNA editing. Some RNAs undergo further modifications and may be transported back to the nucleus for additional processing before they are fully functional. Previous year questions. PYQs 1. CSIR Net Life Sciences 2015. Which of the following RNA molecules is transported from the nucleus to the cytoplasm after splicing and polyadenylation? A rRNA B turn a cmRNA D mRNA. Answer: cmRNA. Explanation: mRNA is processed by splicing and polyadenylation in the nucleus before being exported to the cytoplasm for translation. 2. CSIR Net Life Sciences 2018. What is the primary function of the 5' cap in mRNA export? A protects the RNA from degradation. B facilitates nuclear import. C increases the half-life of the RNA. D assists in ribosome binding for translation. Answer: D assists in ribosome binding for translation. Explanation: the 5' cap is essential for mRNA export, translation initiation, and protection from exonucleases. 3. CSIR Net Life Sciences 2019. Which protein complex is involved in the export of mRNA from the nucleus to the cytoplasm? A TREX complex, BRNP complex, C60S ribosomal complex, D exosome complex. Answer: a TREX complex. Explanation: the TREX complex is involved in the export of mRNA by interacting with RNA and facilitating its passage through the nuclear pore complex. Conclusion: RNA transport is a fundamental process for proper cellular function, ensuring that RNA molecules are correctly processed and transported from the nucleus to the cytoplasm. The regulation of RNA export, particularly for mRNA, is tightly controlled and essential for gene expression. Understanding the mechanisms of RNA transport, the proteins involved, and the regulatory steps is vital for mastering the topic, especially for the CSIR Net JRF Life Sciences exam, where questions may test both basic concepts and complex regulatory details. You said. Protein synthesis and processing. ChatGPT said. Protein synthesis and processing. CSIR Net JRF Life Sciences, maximum detail, paragraph format, no tables, PYQs included with year and answers. Protein synthesis is a fundamental biological process in which cells translate genetic information encoded in mRNA into functional proteins. This process involves several stages, including transcription, translation, and post-translational modifications. The regulation of protein synthesis and subsequent processing ensures that proteins are correctly folded, modified, and directed to their appropriate cellular locations, where they perform vital functions. Protein synthesis occurs in two primary compartments of the cell: the nucleus, where transcription occurs, and the cytoplasm, where translation and protein processing occur. 1. Overview of protein synthesis. The process of protein synthesis is broadly divided into two stages. Transcription. Transcription is the first step in protein synthesis, where the information encoded in a gene is copied into a complementary mRNA molecule. This process occurs in the nucleus in eukaryotic cells and involves several key steps. Initiation. The enzyme RNA polymerase binds to the promoter region of the gene, and transcription factors help in the unwinding of the DNA. Elongation. RNA polymerase synthesizes the mRNA strand by adding nucleotides complementary to the DNA template strand. Termination. Transcription ends when RNA polymerase reaches the terminator sequence, and the mRNA transcript is released. This mRNA is then processed capping, splicing, and polyadenylation before being exported to the cytoplasm for translation. Translation. Translation is the process by which the information carried by mRNA is used to synthesize proteins in the ribosome. This process involves three primary stages: initiation, elongation, and termination. Initiation. The small ribosomal subunit binds to the 5' cap of the mRNA, and the initiation codon August is recognized. The initiator tRNA carrying methionine in eukaryotes binds to the mRNA, and the large ribosomal subunit assembles. Elongation. The ribosome moves along the mRNA, reading the codons. Each codon is matched with an appropriate tRNA carrying the corresponding amino acid. The ribosome catalyzes the formation of peptide bonds between amino acids, creating a polypeptide chain. Termination. Translation concludes when a stop codon is encountered, and the newly synthesized protein is released from the ribosome. Translation in prokaryotes versus eukaryotes. In prokaryotes, translation begins while mRNA is still being transcribed, as there is no nuclear membrane. However, in eukaryotes, transcription and translation are spatially separated, with transcription occurring in the nucleus and translation in the cytoplasm. Eukaryotes also have a more complex initiation process and additional regulatory mechanisms to ensure proper protein synthesis. 2. Post-translational modifications (PTMs). After a protein is synthesized, it often undergoes a variety of post-translational modifications (PTMs) to become fully functional. PTMs are crucial for the regulation of protein activity, stability, localization, and interactions with other cellular components. Some of the most common PTMs include phosphorylation. The addition of a phosphate group to specific amino acids, typically serine, threonine, or tyrosine, is catalyzed by kinases. Phosphorylation can activate or deactivate enzymes, alter protein conformation, and influence protein-protein interactions. It is a major regulatory mechanism in signal transduction pathways. Glycosylation involves the addition of sugar molecules to proteins, which is crucial for protein stability, cell recognition, and interactions with the extracellular matrix. This modification occurs in the endoplasmic reticulum and Golgi apparatus. Acetylation. The addition of an acetyl group to the lysine residues of proteins can influence protein stability, gene expression, and protein function. Histone acetylation is a key process in the regulation of chromatin structure and gene expression. Ubiquitination. The attachment of ubiquitin molecules to proteins marks them for degradation by the proteasome. Ubiquitination serves as a mechanism for controlling protein levels and removing damaged or misfolded proteins from the cell. Methylation. The addition of methyl groups to lysine or arginine residues can regulate protein-protein interactions, protein stability, and gene expression. It plays a key role in epigenetic regulation, particularly in the modification of histones. 3. Protein folding. Proteins must fold into their functional three-dimensional structures to be biologically active. Protein folding occurs in the cytoplasm with the help of molecular chaperones, such as HSP70 and HSP90. These chaperones prevent incorrect folding and assist in the proper folding of proteins, ensuring that they achieve their native conformation. Misfolded proteins can be tagged for degradation by the ubiquitin proteasome system or refolded by chaperones. Chaperones and chaperonins. Chaperones are proteins that assist other proteins in folding without becoming part of the final structure, while chaperonins are large, complex chaperone machines that provide a protected environment for proteins to fold properly. These mechanisms help maintain cellular proteostasis and prevent the accumulation of toxic, misfolded proteins. 4. Protein targeting and localization. Once synthesized, proteins must be transported to specific locations within the cell or secreted outside the cell. This targeting is facilitated by signal peptides and signal recognition particles (SRPs) that direct proteins to the correct compartment. Nuclear proteins contain nuclear localization signals (NLS) that guide them into the nucleus. Secreted proteins are proteins destined for the endoplasmic reticulum or contain signal sequences recognized by SRPs, which help guide them to the ER for further processing. Mitochondrial and chloroplast proteins have specific targeting sequences recognized by receptors on the respective organelles. 5. Regulation of protein synthesis. Protein synthesis is tightly regulated to maintain cellular homeostasis. Several mechanisms control the rate of translation. Regulatory proteins that bind to mRNA and prevent translation initiation. MicroRNAs that inhibit translation or induce degradation of target mRNAs. Translational repressors that bind to the mRNA and block ribosome assembly. Environmental signals such as nutrient availability, stress conditions, and signaling pathways e.g., mTOR pathway can influence protein synthesis. Previous year questions PYQs 1. CSIR Net Life Sciences 2017. Which of the following modifications of proteins is primarily responsible for targeting proteins for degradation by the proteasome? A. Phosphorylation. B. Ubiquitination. C. Methylation. D. Acetylation. Answer. B. Ubiquitination. Explanation. Ubiquitination involves the attachment of ubiquitin to proteins, marking them for degradation by the proteasome. 2. CSIR Net Life Sciences 2016. Which of the following processes occurs first in the translation of mRNA? A. Binding of the ribosome to the mRNA. B. Peptide bond formation. C. Release of the completed protein. D. Binding of the tRNA to the mRNA. Answer. A. Binding of the ribosome to the mRNA. Explanation. Translation begins with the binding of the ribosome to the mRNA, followed by initiation, elongation, and termination. 3. CSIR Net Life Sciences 2020. In protein synthesis, the amino acid is transferred to which site on the ribosome? AA site, BP site, CD site, DS site. Answer. AA site. Explanation. The amino acid is first transferred to the A site, amino acyl site, on the ribosome during translation elongation. Conclusion. Protein synthesis and processing are crucial for the proper functioning of cells and organisms. Understanding the stages of transcription, translation, and post-translational modifications is essential for comprehending cellular processes, regulating gene expression, and maintaining cellular homeostasis. The integration of these processes, coupled with precise regulation, is critical for maintaining protein quality, function, and cellular health. Mastery of these concepts is necessary for excelling in the CSIR Net JRF Life Sciences exam, where both foundational and advanced level questions may be asked. You said, ribosome. ChatGPT said, ribosome. CSIR Net JRF Life Sciences, maximum detail, paragraph format, no tables, PYQs included with year and answers. The ribosome is a complex molecular machine responsible for translating mRNA into protein. It plays a central role in protein synthesis and is found in both prokaryotic and eukaryotic cells. Ribosomes are composed of two main subunits, the large subunit and the small subunit, both made up of ribosomal RNA, rRNA, and proteins. These subunits come together during translation to decode the mRNA and assemble the corresponding polypeptide chains. Ribosomes are present in the cytoplasm and, in the case of eukaryotes, also on the rough endoplasmic reticulum, forming the rougher. 1. Ribosome structure and composition. Ribosomes are composed of two distinct subunits. Small subunit. In prokaryotes, the small subunit is the 30S subunit, while in eukaryotes, it is the 40S subunit. This subunit is primarily responsible for decoding the mRNA and ensuring the correct alignment of the mRNA and tRNA during translation. Large subunit. In prokaryotes, the large subunit is the 50S subunit, while in eukaryotes, it is the 60S subunit. The large subunit is involved in peptide bond formation, catalyzing the reaction between amino acids to form a polypeptide chain. The ribosomal subunits are composed of both rRNA and ribosomal proteins. The rRNA molecules provide the scaffolding for the ribosomal structure and play a critical role in catalyzing the peptide bond formation. The ribosomal proteins help in maintaining the structure and stability of the ribosome. 2. Ribosome function in translation. The ribosome's main function is to carry out protein synthesis by translating the genetic information encoded in mRNA into a specific sequence of amino acids. This process occurs in 3 stages: initiation, elongation, and termination. Initiation. The ribosome assembles on the mRNA molecule, with the small subunit binding first to the mRNA's 5' cap in eukaryotes or the Shine-Dalgarno sequence in prokaryotes. The initiator tRNA, carrying the amino acid methionine in eukaryotes or formylmethionine in prokaryotes, binds to the start codon August on the mRNA. The large subunit then joins the small subunit to form a functional ribosome, which is now ready to begin translating the mRNA. Elongation. During elongation, the ribosome moves along the mRNA, reading its codons. The tRNA molecules, each carrying a specific amino acid, enter the ribosome at the A site amino acyl site. The amino acids are transferred from the A site to the growing polypeptide chain, which is attached to the tRNA in the P site peptidyl site. The ribosome shifts along the mRNA in a process called translocation, moving the tRNA and mRNA by one codon length, thereby allowing the next tRNA to enter the A site. This elongation process continues, with the ribosome adding amino acids to the growing polypeptide chain until the translation is complete. Termination. Translation ends when a stop codon, UAA, UAG, or UGA is encountered. Release factors bind to the stop codon, catalyzing the release of the newly synthesized polypeptide from the ribosome. The ribosome dissociates into its small and large subunits, completing the translation process. 3. Ribosome in prokaryotes versus eukaryotes. Though the fundamental mechanism of translation is conserved, there are significant differences in ribosome structure and function between prokaryotes and eukaryotes. Prokaryotic ribosomes. Prokaryotic ribosomes are smaller, consisting of a 30S small subunit and a 50S large subunit, forming a 70S ribosome when combined. The ribosome in prokaryotes is responsible for both transcription and translation happening simultaneously in the cytoplasm, as there is no nuclear membrane to separate the two processes. Prokaryotic ribosomes are also responsible for translating mRNA that may contain operons, a feature unique to prokaryotic gene expression. Eukaryotic ribosomes. Eukaryotic ribosomes are larger, with a 40S small subunit and a 60S large subunit, forming an 80S ribosome. Translation occurs in the cytoplasm or on the rougher, forming the rough ER, and transcription is separated spatially and temporally in the nucleus. Eukaryotic ribosomes are more complex, with additional regulatory proteins and factors that assist in the translation process, particularly in the initiation phase. 4. Ribosome in organelles. Eukaryotic cells contain specialized ribosomes in mitochondria and chloroplasts, which are similar in structure to prokaryotic ribosomes. These organelle ribosomes are involved in the synthesis of proteins that are essential for the functions of these organelles. The mitochondria and chloroplasts contain their own DNA and use their ribosomes to synthesize a portion of the proteins needed for their respective functions in energy production and photosynthesis, respectively. 5. Ribosomal RNA, rRNA, and its function. The rRNA plays a critical role in the ribosome's function. It serves as both a structural scaffold and a catalytic agent in the ribosome. The large rRNA subunit catalyzes the formation of peptide bonds between amino acids, while the small subunit ensures the proper alignment of the mRNA and tRNA. In fact, the catalytic activity of the ribosome is predominantly attributed to the rRNA, making it an example of a ribosome ribozyme. Previous year questions PYQs 1. CSIR Net Life Sciences 2019. Which of the following is the site of peptide bond formation during translation? A site. BP site. CD site. D ribosomal large subunit. Answer: D ribosomal large subunit. Explanation: the peptide bond formation occurs in the large subunit of the ribosome, catalyzed by rRNA. 2. CSIR Net Life Sciences 2018. The ribosome is composed of which of the following types of RNA? A mRNA. B tRNA D mRNA. Answer: crRNA. Explanation: ribosomes are composed of ribosomal RNA, rRNA, and ribosomal proteins, which together form the structure and functional components of the ribosome. 3. CSIR Net Life Sciences 2017. In eukaryotic cells, translation takes place in which of the following locations? A. Nucleus. B. Mitochondria. C. Cytoplasm. D. Endoplasmic reticulum. Answer: C. Cytoplasm. Explanation: in eukaryotic cells, translation primarily occurs in the cytoplasm or on the rough endoplasmic reticulum, ER, which is studded with ribosomes. Conclusion: ribosomes are essential molecular machines for translating genetic information into functional proteins. Their structure, composed of rRNA and proteins, allows them to decode mRNA, catalyze peptide bond formation, and assist in protein folding. Understanding ribosome function and its differences between prokaryotes and eukaryotes is critical for the CSIR Net JRF Life Sciences exam. A deep grasp of ribosomal function in translation and the regulation of protein synthesis will provide a solid foundation for tackling related questions in the exam. You said. Formation of initiation complex. ChatGPT said. Formation of initiation complex. CSIR Net JRF Life Sciences. Maximum detail. Paragraph format. No tables. PYQs included with year and answers. The formation of the initiation complex is the first crucial step in protein synthesis. It involves the assembly of the ribosome, mRNA, and initiation factors to begin the process of translation. This process is tightly regulated and differs slightly between prokaryotic and eukaryotic cells, though the basic steps remain similar. 1. General mechanism of initiation complex formation. The initiation complex in protein synthesis begins with the assembly of several key components. mRNA. The messenger RNA contains the genetic code that will be translated into a polypeptide. The mRNA binds to the ribosome at the 5' cap region in eukaryotes or the Shine-Dalgarno sequence in prokaryotes. Ribosome. The ribosome consists of two subunits, the small subunit 30S in prokaryotes and 40S in eukaryotes, and the large subunit 50S in prokaryotes and 60S in eukaryotes. These subunits must come together in the presence of mRNA to form a functional ribosome. Initiator tRNA. The initiator tRNA carries the first amino acid of the protein methionine in eukaryotes or formylmethionine in prokaryotes and binds to the start codon August on the mRNA. Initiation factors. These are proteins that assist in the assembly of the initiation complex. They are required to ensure the correct binding of mRNA to the ribosome and the proper alignment of the initiator tRNA. These factors include IF1, IF2, and IF3 in prokaryotes, and ACE eukaryotic initiation factors in eukaryotes. 2. Steps of initiation complex formation in prokaryotes. In prokaryotes, the initiation complex is formed as follows. The small ribosomal subunit 30S binds to the Shine-Dalgarno sequence of the mRNA, a specific sequence upstream of the start codon that helps align the ribosome with the mRNA. Initiation factor IF3 binds to the small subunit, preventing premature association with the large subunit. The initiator tRNA, formylmethionine tRNA, carrying the formylmethionine FMET, binds to the start codon August in the mRNA in the P site of the ribosome. Initiation factor IF2, in the presence of GTP, helps in the binding of the initiator tRNA to the small subunit. If one binds to the small subunit to assist in the proper positioning of the tRNA and prevent incorrect binding of other tRNAs. The large ribosomal subunit 50S associates with the small subunit 30S and GTP is hydrolyzed, releasing the initiation factors. The ribosome is now fully assembled, with the mRNA and initiator tRNA in place, ready to begin elongation. 3. Steps of initiation complex formation in eukaryotes. In eukaryotes, the process of forming the initiation complex involves more complexity and additional factors. The small ribosomal subunit 40S associates with several eukaryotic initiation factors ACE. These include EIF1, EIF1A, EIF3, and EIF5. The mRNA has a 5' cap structure that is recognized by EIF4E, part of the EIF4 complex. This complex also includes EIF4A and EIF4G, which assist in unwinding the mRNA to facilitate translation initiation. The initiator tRNA, MET tRNA carrying methionine, binds to EIF2, which is bound to GTP. The ACE2-GTP initiator tRNA complex associates with the small ribosomal subunit, and the ribosome mRNA complex begins scanning the mRNA from the 5' end toward the August start codon. When the start codon August is located, the large ribosomal subunit 60S joins the complex, assisted by EIF5B, and GTP is hydrolyzed. Once the large subunit is in place, initiation factors are released, and the ribosome is ready to begin elongation. 4. Differences in initiation complex formation, prokaryotes versus eukaryotes. Prokaryotes rely on the Shine-Dalgarno sequence for ribosome binding, while in eukaryotes, the ribosome is recruited through the 5' cap and the ACE. Prokaryotic initiation is faster and simpler, with only a few initiation factors involved, whereas eukaryotic initiation is more complex, involving multiple initiation factors that regulate translation in a highly coordinated manner. 5. Regulatory control of initiation complex formation. Initiation of translation can be regulated at various levels. In eukaryotes, translation initiation is often regulated by the phosphorylation of initiation factors like EIF2, which can inhibit translation under stress conditions. In prokaryotes, regulatory proteins can prevent the formation of the initiation complex by blocking the Shine-Dalgarno sequence or by inhibiting the association of the small ribosomal subunit with the mRNA. Previous year questions. PYQs 1. CSIR Net Life Sciences 2020. Which of the following initiation factors is responsible for binding the small ribosomal subunit to the mRNA in prokaryotes? AIF1. BIF2 CIF3 DEIF4. Answer. CIF3. Explanation. IF3 is responsible for preventing